Hello, my name is Caitlin Raleigh, and myself, along with Nicole Diefenbacher, Rachel Hallman, Megan Kingrey, and Chelsea Reecer, will be presenting the presence of Escherichia coli O157H7 in beef production. Our overall goal is to identify the main sources of E. coli O157H7 contamination in smaller beef cattle operations found in South Dakota. Some of our smaller objectives include: once data is collected that the levels of contamination would be determined based on the time span where the E. coli growth occurred. This will then be used to determine when the level of contamination is too great by presenting one or more colony formations that have occurred in a centimeter squared area that is tested. The problem we wish to solve with this experiment is to determine the origin of E. coli O157H7 contamination in each step of the beef production process. And some background information about Escherichia coli O157H7. This strain of E. coli is gram-negative and highly virulent, requiring very little concentration, fewer than 100 colony-forming units, for a human to become ill in comparison to other strains that may require closer to 1 million CFUs. Most of these other strains are harmless and provide benefits to humans by residing in the intestines, where they create healthy amounts of vitamin K and vitamin B complex. However, a handful of strains similar to O157H7 causes gastroenteritis, which is infectious diarrhea, and produce toxins closely related to the Shigella toxin. These toxins are responsible for causing damage to intestinal epithelium. According to records from the United States Department of Agriculture, contamination requires only one colony-forming unit per centimeter squared on a raw beef carcass. However, ground beef for human consumption may legally contain up to 50 million bacteria per gram of substance. Over 100 outbreaks have been recorded within the past 30 years, 52% of which have been linked to food coming from the beef industry. Data from epidemiologists state that this strain of E. coli may possibly be found in up to 8.3% of the cattle population.
Hi, I'm Nicole Diefenbacher. I will be discussing the beef production process. Production begins with the cow-calf operations. These include cows being bred in a timely manner in which they give birth to a calf once every year. Calves will then spend the first few months of their lives here with their mothers and survive strictly on the consumption of milk and whatever grazing is available to them. From the cow-calf operation, these calves are weaned from their mothers around the age of 7 months. Calves should weigh approximately 500 to 600 pounds. At this point, they are weaned and are introduced to grass for grazing purposes only. From weaning, they are moved into feedlots in which mature calves are sold and moved here in order to further their growth and development. These cattle have continuous access to water and feed, which has been carefully balanced for this point in their lives. Cattlemen work with veterinarians and nutritionists on a daily basis in order to ensure the best care for each animal, while also maximizing the amount of growth and gain. The next step in the beef production process is manufacturing. This entails the packaging plant. Between 18 and 22 months of age, cattle should weigh between 1,200 and 1,400 pounds. At this point, they are sent to processing facilities where they are harvested and their carcasses form meat products such as steaks, roasts, and ground beef. The USDA places many inspectors at this part of the process in order to make sure things are running as they should and focus on keeping contamination of meat product to a minimum. From the manufacturing, there is distribution and delivery. This entails the food service and retail industry. Operators within this process follow many steps in order to provide safe and healthy products to the consumer. Vehicles should be constructed to protect the product and should be sanitized properly between every load to be transported. Care should also be taken when loading and unloading shipments. Also, products should be checked continuously throughout transport in order to ensure safety precautions are being followed at all times. Preparation and consumption in restaurants, supermarkets, and homes is another key element of the beef production process. The USDA recommends most beef products to be cooked at a minimum of 145 degrees Fahrenheit. However, ground beef mixtures, such as hamburgers, as well as organ meats, such as heart, kidney, liver, and tongue, should reach an internal temperature of 160 degrees Fahrenheit before consumption.
Hi, my name is Rachel Hallman, and I'll be talking about the methods of our experiment. We have decided to test samples of beef from each stage of production because we want to get an idea of where the E. coli is coming from for each stage. We decided to focus on a smaller population of cattle so that it won't be as large numbers and we'll have more accurate data. We decided about 500 to 1,000 head per farm. We picked a location that is not in a high producing state. We picked South Dakota because they have less strict guidelines of production, so it would be easier to find a flaw in the system. We are hoping that the research that we find will enable an adjustment in the food safety policy to better serve clients. We will be taking samples from 5 different stages. We will start at the feedlot where the cows are raised, and we'll take a sample from them. And then we will take another sample in the slaughter phase where the cows are being prepared and to be processed, which is the 3rd stage where we will take another sample. And then we'll take a sample at the shipping phase where the cows are being sent off to the markets. And lastly, we will take a sample at the stores where the consumers will be picking up the meat to take home for consumption. The samples will be tested using a variety of different methods in the laboratory. We will be using different slide stainings and plate pouring techniques to determine if E. coli is present or not. And then we will compare the bacterial counts of what we found in the lab to the U.S. health standards to see where they stand. Further investigation into the stages will show if there are outstanding bacterial counts in those areas. And once we determine the cause, we will be able to look into a preventative method to stop the spread of E. coli. And if we have identified a cause or a source of E. coli, we will try to implement a different tool or procedure or process to decrease the bacterial counts. After the tool or process have been implemented, we will look at the bacterial count again, and we will determine by an increase or decrease if our method has worked. An increase shows that the process did not work and that we have actually worsened the problem with E. coli, and a decrease shows that our tool or process has helped decrease the number of E. coli and therefore making the product safer for human consumption and helping to eradicate the E. coli in the industry.
I'm Megan, and I'll be talking about the timeline of events for this project. This project can be performed at any time of the year, as long as there's no drastic environmental changes at the time. It'll start as soon as you pick a group of cattle and follow it from the stockyards all the way to the store. We're choosing to select samples during the summer months in July and August. Once the cattle go into slaughter during the fall, the carcasses will then be sampled, and those samples will be taken to a laboratory for testing. Once the carcasses have been slaughtered, they will be tested again just before they're packaged. At the end of the transportation to the seller, either a private seller or a retail operation, the meat will again be tested before it enters the store. After the meat has been on the store shelves for 24 hours, it will again be tested and the samples will be taken to the lab. Once the data has been collected, it will be analyzed and can be put into a graphical format in order to illustrate any relationships that there are between the stages of production and the levels of bacteria. After the data has been analyzed, a certain tool would be implemented in order to lower any excess bacteria levels. After 1 month of implementing this tool, the meat will again be tested through all the same processes, and the final data will be analyzed in order to determine whether the outcome of that tool was successful or not.
Hello, my name is Chelsea Reecer, and I will be doing our dissemination of data, results, budget, and future goals. So the dissemination of our data and results, data will be spread by informing farmers, meat packagers, and consumers about the presence and danger of E. coli strand O157H7. Information given on prevention measures that may be enacted to prevent problems in the future will be given to all of the above stated receivers, and studies will be posted online where the pertaining parties will be able to have access to broaden their knowledge base. As far as our budget is concerned, most research is able to be done on a farm or lab setting, eliminating the need to transport the animals. Obviously, transporting animals would be an excessive cost that we have hence avoided by doing our research on those two separate playing fields. Fuel prices will actually need to be considered a little bit for researcher travel. So if we hire outside researchers or our team needs to get somewhere, we will have to factor in fuel prices, and obviously those vary every day. Laborer pay may also be necessary due to a few people being required to herd cattle into proper positions. If we put them in a headstall, somehow we need to have people move them into the correct position. So we will need at least a few people other than ourselves, because obviously we'll be taking data and doing other things. We need someone to get the cattle wherever they need to be. And our budget for our lab equipment is relatively conservative, I think. Glass slides we'll need for spreading samples and looking at them under a light microscope. And those run about $10 for a good amount. Light microscopes are a little bit more pricey. Those are, if you get a basic one, can be $200 is what we estimated. It can go up to like $500 depending on the quality of the microscope, but we went with the average, so about $200 for that. Petri dishes, depending on the quality of the Petri dish, can be very cheap or very expensive. And we went with an average type of Petri dish, average size. So that is going to be, for the amount we need, is going to be about $900. So we're going to be going through a lot of Petri dishes. If we have mistakes or spoiled results or negative results that we weren't looking for, obviously we'd have to redo those and we would need a new Petri dish. So just to overstock and be prepared, we planned for $900 spent on Petri dishes. Standard growth augers, such as, but not limited to, McConkey's auger, mannitol salt auger, eosin blue auger, and tryptic soy broths. These are all very general augers that can be used in any lab facility for a variety of purposes. That most of them are selective for a specific gram stain or maybe a specific kind of bacteria. So we ran the numbers for those, and McConkey's auger tends to be about $200 for the amount we would need, and mannitol salt auger would be about $800. Eosin blue would be another $800, and tryptic soy broth as well would be about $800. So totally, it would be about $2,600 just on auger, which is one of the most important factors of our experiment because that's when we'll be doing all of our growth on the auger plates. We would also need various slide stains to illustrate the qualities of the meat we sample. So crystal violet, carbol fuchsin, methylene blue, safranin, Graham's iodine, those are just a small portion of the various dyes we could use. Those are the dyes we selected to try. So the Graham's iodine and safranin and crystal violet can all be used for Graham staining. There's a variety of techniques you could do with all of these slide stains. So crystal violet turned out to be about $60, carbofuchsin about $150, methylene blue about $60 again, and safranin was about $100, and Graham's iodine is about $120. So dyes are just as important as the rest of the equipment, if not more so. Next up, we have decolorizing agent, which is another tool used in Graham staining. So we would use a decolorizing agent such as 95% ethanol, very general, very easy to obtain, and that would run about $60 for the amount that we would try to contain ourselves to. Then we have some general equipment that isn't necessarily lab specific, like markers. So markers obviously very cheap. We got some nice ones like $7. And then cover slips are about $20 for a lot, like 1,000. It varies on the quality and the brand of the cover slips. So we did $20. We allowed $20 for cover slips. Bunsen burners. Again, it's just like the microscope. It can vary in size and quality and power. So about $40 is what we allowed ourselves for Bunsen burners. A spreading tool. So when we have our sample and we spread it on our actual slide or our petri dish to observe and to grow in culture, we would need one of those, and those are typically very cheap, either $1 to $5 depending on the material. It could be like a plastic spreading tool, or it could be like a glass one or a metal one, so it really depends on the material that the spreading tool is made out of. An incubator. So when we grow our cultures on our petri dishes, we are going to need an incubator to allow them to grow at the right temperature. To remain constant, to get constant results, and to make sure all our results are accurate. So we would need an incubator for this experiment because we need to assure that out of all the samples we're taking, we are staying constant at the right temperature for growth. So an incubator runs about $500. Depending on size, you can get one that's the size of a refrigerator, or you can get one that's the size of like a shoebox. So you really have to kind of play into that factor. So we chose one that was medium size, about the size of a mini fridge. So about $500 for that. And lastly, we have our autoclave, which autoclaves are extremely expensive, but again, they're an invaluable tool in the research process. They're used for sterilization. It's one of the most effective ways to sterilize things and ensure aseptic technique. And we allowed ourselves about $4,500 for that. So we would get an autoclave just to be on the safe side, so to make sure we don't have any cross-contamination of bacteria. So our preliminary total for our budget without labor costs is approximately $9,332. Now, factoring in labor costs, obviously things may vary depending on how many people we need, but we allowed ourselves $500,000 total, including tools and labor costs. Our future goal in this study is we hope to identify the sources of E. coli O157H7 contamination, and also further the study and investigation of this subject, and this may be conducted in order to implement a surveillance program in order to control the level of contamination in meat supply. And that's our presentation, and I hope you enjoyed it, and I hope you found it very informative, and thank you for listening.